Okay, so welcome back. I hope you have enjoyed your break. The video was getting long, so I thought I'll split into two parts. Without much further ado, let's start modalities for treatment. So I've enumerated all the modalities that I could find. So there's gene therapy, recombinant protein therapy, transfer of corrected grafts, autologous epidermal sheets with the corrected gene editing by using TALEN and CRISPR. So these are two ways in which gene editing can happen. There's also another way known as zinc finger motif, but we will not discuss. It's the first method of uh, zinc finger gene editing. It was the first method of gene editing, now not used. Now CRISPR is in fashion. So TALEN and CRISPR are gene editing. Intradermal allogenic fibroblast, bone marrow derived stem cells, mesenchymal stem cells, and Revertant Mosaicism. So we'll go through each of them very briefly so that we can cover all the modalities for treatment. Now remember, these are not modalities for, they can be used for management, because if a child has EB and chronic non-healing erosions and ulcers, some of these modalities can be used to treat those ulcers, treat those erosions, and even decrease the chances of patient developing newer erosions, at least theoretically. Okay, so these are for treatment of the disease. Initially in part 1, we had discussed management of the disease or management of complications. Okay, so let's go through each of them one by one. First is recombinant gene therapy. It is very easy to understand. Recombinant gene is translated in patient with mutated gene. For example, let's say you have a gene here. Let me just draw one gene. Okay, and one of the allele is mutated. So what happens is, uh, let me just make it again. So this is normal DNA, this is normal DNA, and just one part of it has the mutated gene. What you can do is you can cut it here, remove it here, and then supply them the corrected gene. Supply the patient to the corrected gene, and the cell will automatically put the gene here and repair it. So this is gene therapy, and since you are using a recombinant gene, a gene that you have created in a lab, recombinant gene, it is known as recombinant gene therapy. Okay, clear? You take out the affected mutated gene, replace it with a newly formed in vivo created or lab created gene, and attach again, and you have the problem is solved. It is as same as when the bulb gets fused, you remove the bulb, put in a new bulb, and everything is okay. That is recombinant gene therapy. One good example is the LAMB3 transgene in junctional EB, in which you take out the mutated gene, you put a new LAMB3 which is perfectly normal, and it has been shown in a case report to have at least 80% correction of skin and intermediate junctional EB. So it does show a lot of promise. Now, keratinocyte stem cells can be also used. What you do is you actually take out keratinocytes, you convert them into stem cells by, of course, a very complicated process. In those keratinocyte stem cells, you take out the genetic material, take out the bad gene from it, insert good gene, insert the corrected genetic material inside the stem cells, and then re-implant those stem cells inside the erosions or the ulcers. And then the new keratinocyte stem cells will start to grow, and they will not have the mutation, and the child will be cured. Clear? Many of the time, keratinocytes can be combined with fibroblasts, and fibroblasts with corrected collagen 7A1 gene. Now remember, keratinocytes are the main cells in Epidermolysis bullosa, at least the simplex variant. Fibroblasts are mainly in the dermis, and fibroblasts secrete a lot of proteins, and an important one regarding basal membrane zone is collagen 7. So what you do is you take out a sample from the patient, you take out the genetic material from patient's fibroblasts, correct the gene, put it back again, and let them grow. And now they are growing with the proper gene. They will not show the disease in the child. Clear? So collagen 7 is being used in the dystrophic EB because that's where the mutation actually lies. So this diagram actually tells you that what all forms of genetic editing can be done. Either you can replace the gene entirely, which is gene replacement. For example, you take out the nuclear material, take out the gene, put a new gene and put it back again. Or CRISPR, We'll discuss CRISPR in one slide, how it works. RNA based. So instead of putting the gene, you put the RNA which encodes for the protein so that new protein, proper protein, natural protein can form. Or Revertant Mosaicism, which we'll discuss in one slide, don't worry. So what actually you do is you take out keratinocyte, you take out fibroblast, you correct the gene correction, you then ask them to multiply. Okay, you allow the cells to multiply so that they become graftable, and then to transplant that graft on the patient. So that is gene therapy. In recombinant protein therapy, you directly supply the protein. So what exactly happens is, let's say you have a gene, this will form an RNA, okay, RNA, this will form an mRNA, and this will form a protein. So in gene therapy, you are actually supplying the gene. But in recombinant protein therapy, you form the corrected protein inside the lab and directly supply the protein. Okay. So for example, in you have to make some food. Okay, you have to make a food. Let's take example. You have to make biryani, and you while making biryani, you don't have the correct recipe, and the biryani is bad. So in gene editing, we give you the corrected recipe. We give you the correct recipe so that you can make proper biryani. While in the recombinant protein therapy, we get biryani properly made for you and you just replace your old biryani with the new corrected biryani. Clear? So in protein therapy, you actually just replace the protein instead of replacing the gene. So you supply recombinantly made protein to a patient which has mutated protein. Clear? So in protein therapy, proteins are exchanged. In gene therapy, genes are exchanged. Topical treatment was studied but not found to be very beneficial because the erosions have infections and proteases that actually destroy the recombinant protein. Systemic agents are under trial, especially for collagen 7 in dystrophic EB. As of now, they are just under trial to find out whether systemically you can give proteins and that can help to improve the life of a child with EB. TALEN. Now, TALEN is a method of gene editing. So while writing a short note, you just have to mention that TALEN and CRISPR-Cas9 system can actually be used to edit the genetic material. So TALEN is the short form of transcription activator-like effector nucleases. Since these are nucleases, they will cut the DNA. So in this diagram, this is a DNA strand, okay, double-stranded DNA. What TALEN does is it recognizes a pattern, it recognizes a sequence of nucleotide, attach itself to it, and then cut it at a predisposed location. So a double-stranded DNA is cut by TALEN at a location where the mutation is present, and then you supply the corrected gene which gets incorporated. So what gene therapy was the process, TALEN is the tool. Clear? So TALEN cuts double-stranded DNA pre-programmed base pair and cut areas repaired by adding the corrected gene. The gene gets replaced and the patient is cured. Clear? Let's move forward. Now, CRISPR-Cas9 system is also a method of gene editing and it is latest as compared to TALEN. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats plus CRISPR-Associated System. So CRISPR full form is this, Cas is CRISPR-Associated System 9. Clear? Now what it does is, just like TALEN was recognizing nucleotides and cutting at a place, CRISPR also does the same thing. CRISPR has a guiding RNA, gRNA, and guided RNA actually scans the DNA molecule and attach to a pre-decided location. Okay, whatever the location is, we decide. The genetic engineers decide that I want to cut DNA at that place. Okay, so here the guide RNA will actually scan the DNA till it reaches the location and stick it there. While the DNA sorry, while the DNA cutting domain will cut it at this location. Okay, so what actually happens? It works same as TALEN. The only advantage is that CRISPR is more precise. So the problem with any inadvertent cutting is not there. You can actually scan and actually cut it at a proper place. So CRISPR-Cas9 system is actually molecular scissors with guide RNA in between. It cuts gene, at a desired point. The cut area is again replaced by adding corrected gene. The gene is replaced, everything is hunky dory, everything is good. Okay, so you can use two systems to do gene editing known as TALEN and CRISPR. Okay, let's move forward. CRISPR is a hot topic. I think I've been asked in Viva once what is the use of CRISPR in dermatological disorders. So this can be used in all genetic disorders theoretically, like xeroderma pigmentosum. Okay, even incontinentia pigmenta, all those sorts of genetic disorders can easily be tackled if you just remove the bad gene and replace it with the good gene. Now, transfer of skin grafts. So what actually happens is you take out the cells from the patient or from some other individual, the donor. The cells can be either keratinocytes or fibroblasts or dermal components. You multiply the cells, okay, and then you form a graft. And that graft can be implanted on the erosions or ulcers so that they can heal properly. So these are known as transfer of skin grafts. You can have autologous split thickness. Autologous means from the same patient. For example, let's say I'm a child with suffering from EB. I have erosions here, and this I don't have erosions. And never have I ever developed erosions here. So the graft will be taken from here and then transferred to cover the erosion on this side. So that is known as autologous. Autologous means same body. Okay, split thickness graft. Allogenic means same species. So, for example, another person can donate their skin cells, their skin graft, and those grafts can be implanted on my erosions. Okay. So, autologous split thickness has short-term benefit, while the allogenic keratinocyte grafts can be useful in managing pain. There are skin bioequivalents, which are lab-created grafts. Okay, so these are not natural; these are lab-created grafts, and they have been shown to have mixed results. Okay, lab-created with mixed results. Okay, let's understand them better. So in the previous slide, we discussed the use of keratinocyte cells to form a graft. Here we'll discuss the advantage of using fibroblasts. Now, fibroblasts are important for maintenance, production, remodeling, and homeostasis of the extracellular matrix, which is the predominant component of the dermis. So in the dermis, that is where the majority of fibroblasts are there. And because of that, it is majorly involved in maintenance, production, remodeling, homeostasis. Okay. Fibroblasts express components for the basement membrane, but mainly collagen 7, which is this is very important in the setting of dystrophic EB. Okay, so these are very important. So they can generate multiple collagens, but collagen 7 is the main collagen. Many a times, fibroblasts are combined with keratinocytes so that you can actually influence the basement membrane formation properly. It has been found that fibroblast injections can actually help in re-epithelialization of chronic erosions. So what actually happens is that you take fibroblasts either from the patient or another human donor, and then you culture them while after changing the genetic problem, genetic mutation, you culture them, make a graft, and inject them around the erosions so that it can help in modeling of the extracellular matrix and the basement membrane formation so the ulcer heals properly. The only problem is that the injections required are repeated and the injections are painful. So we have to keep that in mind. So just to recapitulate, what do we mean by autologous and allogenic? In autologous, you have the patient here. You take a skin biopsy. From the skin biopsy, you take out the fibroblast. You correct the gene. You can correct the gene by gene editing, which can be done by TALEN or CRISPR. After you removing the mutated gene, you put the correct gene again, you expand them. Expand means let them proliferate, let them form a mesh. Then you form a graft and then you implant it back. If the source and the destination is the same individual, it is autologous cell therapy. If the source is different, it's a healthy donor, then it is allogenic cell therapy. But the steps remain same. From a healthy donor, you take a skin biopsy, you isolate fibroblasts. Since the healthy donor doesn't have a mutation, you just have to expand them and then implant them. Okay? So now we are clear about autologous cell therapy and allogenic cell therapy. Let's move forward. Coming to stem cells, I won't diverge much here. In stem cells, what you do is you first take out some stem cells from the bone marrow, then you irradiate them, then you use gene editing to correct the problem. After gene editing, you convert them into skin grafts or mesenchymal stem cells, and then you again implant them on the erosions. Okay, same thing as the fibroblasts, but here the fibroblasts are replaced by stem cells. Depending on the source of stem cells, it can be bone marrow. So bone marrow stem cells, you can have adipocytes, in which you have adipocyte-derived mesenchymal stem cells. You can have umbilical cord mesenchymal stem cells. And this is from the umbilical cord. Lot of hot topic regarding source of stem cells. Maybe time permits, I'll make a video sometimes in the foreseeable future regarding the use of stem cells in dermatology. Maybe if a lot of people demand for it. So this is how stem cells can be used. Again, what you're doing is you're taking out the cells, you are inducing them so that they can go back and become stem cells. You correct the gene modification, you put the corrected gene, you put them back, and then they will start proliferating as normal cells. Revertant Mosaicism. Now, this diagram is a very good diagram. So pause the video, go through this diagram. You will easily understand the concept of revertant mosaicism. Okay, but just understand the concept very quickly. In certain areas of the body, so here you can see in this diagram, it is showing a revertant mosaic patch. So what actually happens is it has been found in evaluation of children of EB that some areas do not show erosions or ulceration even as the time progresses. It has been postulated that these cells which give rise to that particular area have somehow dodged the mutation. They have somehow bypassed the mutation. Okay, and now they are behaving normally. And this can happen in two ways. Either the mutation got cancelled or didn't happen, or they had a separate mutation that is negating the effect of the first mutation. Let me explain it again. Let's say you have AGA as a nucleotide pair, and this leads to protein X, and this is what you know. This is what you need normally. But this gets changed to ATA. Now it's giving you a protein Y. Okay. So what happens is, let's say if this mutation happens, the child has AB. If this mutation happens, but in case of revertant mosaicism, this ATA undergoes again a mutation and turn back to G. Or it can have another mutation and turn back to C, while ACA also translates for X. Okay, so you have mutation and another mutation that negates the effect. And because you have an area of the body which doesn't have the clinically presentable mutation, that area would behave just as a normal skin would. So you can use that area as a source of normal cells. So in a patient of, let's say, autologous transplantation, you can take samples from the unaffected area, culture it, make a graft, and then put it back on the affected eroded area. So that is the concept of revertant mosaicism. Write down in the comments if you don't understand it. It's a very interesting concept to understand, very important also. But apart from academic interest, it's a very important concept to understand in genetic disorders, the concept of revertant mosaicism. Let's move forward. Small molecule therapy, this is only for academic interest. Let's say your Viva is going exceptionally good, so you might reach here. Small molecules are molecules with less than 900 molecular weight. They can pass across cell membranes freely and then they interfere with the target molecules. I'm just going to mention the names which are mentioned in Rooks 10th edition. So gentamicin have been used along with topical botulinum, topical diacerin, oral sulfopitant, not along with but separately to decrease the itch associated with inflammation in EB. Epigallocatechin 3 gallate have been used to decrease the number of blisters. Coenzyme Q topically have been used to improve wound healing. Intradermal Botox has been used to plant for treating of plantar pain and blistering. Also, it decreases sweating. So, as we know, blistering increases because of sweating, so that can happen. Pregabalin decreases pain and itch. Topical sirolimus, which is an mTOR inhibitor, has been used to manage EV simplex associated keratoderma. Oral or IV rigosertib, a polo-like kinase 1 inhibitor, has been used to manage SCC. I have no clue about this. Maybe I'll read further and explain, but trust me, this is going too far. Everything you need to know how to manage a case of EB is in this video before this, okay? Topical ropivacaine for decreasing the wound pain. Clear? So we won't spend much time on this slide. Now, I think this is the last slide, skin carcinomas. Then we have DEBRA and then we have finished. Skin carcinomas are very common, especially SCC, and they are particularly very aggressive. More aggressive as the normal SCC that you see. Let me just write it properly. Very, pardon my bad handwriting. Very aggressive. Yeah, that's the best I can. So the incidence is roughly about 7 to 70% of the patients, especially patients of recessive dystrophic EB, can eventually have SCCs. So it is recommended to do skin surveillance from 5 years of age. It can be annual initially, but better recommendation is every 3 to 6 months. Use photographs and look at the sites of bony prominences or areas of scarring or delayed healing. And you keep on evaluating these sites because these are the sites which are most prone to developing SCC, acral areas and the bony prominences. If you have any suspicious lesions, you biopsy and rebiopsy. What do we mean by that? You do biopsy, and if you miss any kind of malignant finding or any kind of carcinomatous findings, you might again take a biopsy if you're still suspicious. Let's say you have a lesion, you took a biopsy, it was okay, it didn't show any SCC features, but after 6 months it is showing certain change in architecture, then do again a biopsy. Okay, it's very important to catch SCC early on. If the lesions are inaccessible or large, okay, or at a site which is not suitable for biopsy, consider magnetic resonance, MR or CT. Okay, consider that. Chemotherapy and radiotherapy can be used, especially for debulking, but they're found not to be much beneficial. Surgical treatment like deployment of the region, uh, mild local excision, even amputation has been recommended. Okay. EGFR receptor antagonists or immune checkpoint inhibitors have shown some impact, but the research is going on. These are newer modalities. As I've said, SCCs in the setting of epidermolysis bullosa are particularly aggressive. Distant METs have been reported in about 38 to 40% of patients. So make sure you do PET scan to evaluate the spread of the disease in a suspected case of SCC in EB. Okay, so this is the last slide of the presentation. I think so. I think nothing is there in front. And this is about DEBRA. So DEBRA is a non-profit organization. It's a charitable organization. It is set up in various countries. I think more than 40 countries have DEBRA set up. And DEBRA is actually name of a child. Okay, and after the name was kept as DEBRA, it was retrospectively changed as an acronym. So retrospectively, that means at the time of founding DEBRA, this was not the name, but now it stands for Dystrophic Epidermolysis Bullosa Research Association. So that is DEBRA. So DEBRA is international medical research charity dedicated to treatment and cure of epidermolysis bullosa. Treatment and cure. They have a tagline because the cost of doing nothing is too great. So it funds researches. It has just go to the site of DEBRA. It has wonderful resources in managing EB patient, wonderful posters, wonderful summary of each kind of EB. Okay, it has all the sources for EB, education, charitable help, medical management research, everything is there. And I'm talking a bit about DEBRA because I wanted people to know about DEBRA. And also it has been asked once in the Viva to me. So I thought that one should know what DEBRA is. And they also have a tagline, worst disease you have never heard of. So this can be a very good, you know, quiz question. Which disease is a disease that you can think of has a tagline, worst disease you have never heard of. So that's EB. Okay, so DEBRA, you can see the butterfly here, butterfly skin. So with that, I finished this video. It's a very long video, so I decided to split into two parts. I'll edit everything, and if time permits, I'll upload both the parts together. Even if I'm not able to do it, maybe I'll upload it next day, but I think most likely both parts will be uploaded together. Now, these are some reading recommendations. The chapters on genetic bullous disorders are again the same. This article highlights the use of dermal fibroblasts. If you want to know more about how fibroblasts can be used in treatment of EB, go through this article. This way has a good summary of all the different modalities to treat EB, and this is for the tissue engineering skin grafting. So what actually happens is you take a cell, you correct the gene problem, then you provide a scaffold or a framework, an artificial framework, a lab created framework, and you transfer the cells to that framework, so they will form a skin or they will form a membrane, and then this membrane gets transplanted over the erosion. This is a very good tissue engineering thing that will require less amount of cells and more amount of erosion. You can have larger grafts so that larger erosions can be treated. A very good article, you should go through it. Okay, let's move forward. So with that, we finish. I think this is the sixth video on Epidermolysis bullosa. If you go through these six videos, simplex junctional dystrophic diagnosis of EB, and this approach and management to epidermolysis bullosa, I think you have covered whatever you need to know about genetic bullous disorders. Although I've tried to be as explanatory or as easy to understand. And if you combine this series with the basement membrane zone series, your concepts about basement membrane zones will be crystal clear. You will have no problems in remembering the order of proteins, where the defect lies. And if you have gone through these videos properly, you will understand that why this particular defect is leading to this particular clinical manifestation, why you see more scars and granulation tissue in junctional while more scars, less granulation tissue in dystrophy. So I've tried to cover. EB is a tough disorder to understand because we don't give time, we don't have access to patients so that we can actually see the clinical outcomes. But if we are careful enough, so we won't miss diagnosis of EB. There have been instances where people have been diagnosed with EB simplex in their adulthood, in their adolescence. So we should not miss, while keeping in mind the differential diagnosis that we discussed in the first, second, or third slides. That what are the differential diagnosis of EB or blistering in a neonate. So we have to keep that in mind, especially in a developing nation like us. So I won't take much time. This is already a long video. I hope you enjoy your weekend. I hope I've made up for the last week's absence so that I can make a longer video. And thank you for seeing. Any suggestions, you should comment your doubts, queries. I I do take time in replying because a lot of comments and queries come. But you feel free to comment. You feel free to give suggestions. I do reply to you. Those of you who have commented know that I reply to them. Maybe I'm a bit late, maybe I'm early, but I do reply to them. So without much further ado, adios and bye bye. Enjoy your week.